Melatonin does not induce sleep. It terminates the biological day. When this molecule surges into circulation, it issues a system-wide mandate. Stop producing, start repairing. Cells that were synthesizing proteins shift to quality control. Organs that were responding to external demands redirect energy to internal maintenance. The autonomic nervous system pivots from sympathetic dominance to parasympathetic recovery. Core body temperature drops. Blood pressure falls. The liver stops processing incoming toxins and begins regenerating hepatocytes. This is not negotiation. It is a forced state transition. When the melatonin signal fails, when it arrives late, weak, or not at all, the body remains locked in production mode during the hours allocated for repair. Metabolic byproducts continue circulating. Inflammatory cytokines remain elevated. DNA damage accumulates. The cost is not subjective fatigue. The cost is measurable cellular breakdown. Deep in your brain sits a cluster of approximately 20,000 neurons called the suprachiasmatic nucleus. Your master clock. It does not count time. It tracks the Earth's rotation by monitoring light. Your retina contains two separate detection systems. The first, rods and cones, builds images. The second, discovered in the early 2000s, consists of intrinsically photosensitive retinal ganglion cells. These cells measure ambient light intensity and wavelength. They report directly to the suprachiasmatic nucleus. In 2008, George Brainard's laboratory at Thomas Jefferson University published the action spectrum of human melatonin suppression. They exposed volunteers to monochromatic light across the visible spectrum and measured plasma melatonin throughout the night. The data revealed peak sensitivity. Blue light between 446 and 477 nanometers suppressed melatonin with 5 to 10 times the potency of any other wavelength. This is the exact spectral signature of a cloudless midday sky. For 300,000 years, this wavelength meant one thing: the sun is up. The circadian system evolved to use this signal as an unambiguous temporal anchor. It assumes blue light equals day. It does not distinguish between sunlight and a screen. LEDs changed everything. Modern white LEDs are blue LEDs coated with yellow phosphor. Under the surface, they emit a massive spike of energy in the 450-480 nanometer range, exactly where your circadian photoreceptors are tuned. In 2011, Catherine West and George Brainard's team tested this directly. They exposed healthy adults to blue LED light at controlled intensities and measured melatonin suppression in real time. The response was dose-dependent and brutal. At 600 microwatts per square centimeter, melatonin was nearly obliterated. The half-maximal effective dose, the light intensity required to suppress 50% of melatonin, was just 19 microwatts per square centimeter. A typical smartphone held at normal viewing distance delivers between 30 and 50 microwatts per square centimeter of blue-weighted light directly into your retina. The Lighting Research Center in Troy, New York, quantified the circadian threshold in 2015. They measured light in melanopic lux, a unit that weights light by its effect on the circadian system. Below 10 melanopic lux, melatonin remains intact. At 47 melanopic lux, suppression reaches 10%. At 50 to 100 melanopic lux, the range delivered by a phone or tablet in a darkened room, melatonin drops by 15 to 36%. A 2011 study in applied ergonomics tested 2 hours of evening exposure to an LED backlit tablet. Melatonin suppression, 55%. Onset of dim light melatonin, the physiological marker of biological nightfall, delayed by approximately 90 minutes. Your suprachiasmatic nucleus receives a blue light signal at 10:30 p.m. and interprets it as solar noon. The shutdown command is canceled. The body remains in alert configuration. The repair window shrinks before it even opens. The failure propagates through every system that depends on circadian timing. During wakefulness, neurons consume glucose, fire action potentials, and generate metabolic waste. Two proteins accumulate: beta amyloid and hyperphosphorylated tau. Both are causally linked to Alzheimer's disease. The brain has no lymphatic vessels. It relies on the glymphatic pathway, which operates almost exclusively during deep, slow-wave sleep. Cerebrospinal fluid flows through perivascular channels, flushing interstitial waste into the venous system. Melatonin is the key that activates this process. A 2013 review in PMC titled Melatonin in Alzheimer's Disease documented the mechanism. Melatonin directly inhibits beta amyloid generation, prevents fibril aggregation, and arrests TIU hyperphosphorylation. When researchers experimentally suppressed melatonin synthesis in animal models, TIU phosphorylation increased. Spatial memory collapsed. A 2023 paper in PMC confirmed this in human-relevant models. Melatonin administration reduced beta amyloid accumulation and improved short-term memory. The authors noted that melatonin levels are significantly lower in Alzheimer's patients compared to age-matched controls. Suppress melatonin, compress deep sleep, reduce glymphatic clearance. Proteins stay in tissue. The aggregation threshold is crossed. Plaques form. This does not happen in one night. It happens across 10,000 nights. Every cell sustains DNA damage daily. Ultraviolet radiation, reactive oxygen species, replication errors, all generate lesions in the genome. Repair systems exist. They require downtime. In 2015, researchers at UCLA published a study titled Partial Sleep Deprivation Activates the DNA Damage Response. They restricted sleep in older adults to 4 hours for a single night, then analyzed gene expression in peripheral blood cells the following morning. One night of truncated sleep activated the DNA damage response pathway. It upregulated P16 idunat tin ink for harli, a marker of cellular senescence. It increased expression of the senescence-associated secretory phenotype, a pro-inflammatory program that accelerates tissue aging. The authors concluded, sleep loss promotes biological aging through the accumulation of unrepaired damage. After one night, a 2025 study examined Mexican cavefish that evolved to sleep significantly less than surface-dwelling relatives. Despite millions of years of adaptation, these fish still exhibited elevated levels of GH2AX, a marker of DNA double-strand breaks. Even an evolutionarily optimized system cannot escape the cost of insufficient repair time. Your genome does not have infinite redundancy. Each unrepaired break, each persistent oxidative lesion, each cell that enters senescence instead of repair, this is the ledger. It accumulates until the system crosses a threshold and the damage becomes a disease. Melatonin is an oncostatic agent. In laboratory models, melatonin inhibits tumor cell proliferation, disrupts angiogenesis, and modulates clock genes that regulate the cell cycle. A 2009 review in Science Direct titled Melatonin, Sleep Disturbance, and Cancer Risk synthesized decades of epidemiological data. Breast cancer risk was up to 5 times higher in industrialized nations with widespread artificial light at night. A 2004 study in Nature asked, does light at night compromise physiologic cancer protection by suppressing melatonin? The answer was unambiguous. Yes. A 2025 paper in PMC documented how melatonin prevents tumor growth through immune modulation and circadian gene regulation. It enhances natural killer cell activity, increases T-cell-mediated cytotoxicity, and suppresses pro-tumorigenic inflammatory pathways. Night shift workers provide the human data. A 2005 study measured light exposure and melatonin levels in shift workers versus day workers. Night shift workers exposed to just 192 lux during their nominal sleep hours produced 33. 8% less total melatonin over 24 hours. The health outcomes tracked the hormone levels, significantly higher rates of breast cancer, colorectal cancer, prostate cancer, and endometrial cancer. Chronic melatonin suppression removes a layer of cancer defense. Mutations occur at the same rate. Surveillance weakens. Abnormal cells get more opportunities to evade detection. Tumors gain time. Glucose metabolism operates on a circadian rhythm. Insulin sensitivity peaks in the morning and declines at night. This is hardwired into pancreatic beta cells, hepatocytes, and skeletal muscle. Melatonin enforces this rhythm. In 2016, researchers published a study in PLOS One examining the metabolic effects of blue-enriched light exposure. They exposed healthy adults to 30 minutes of blue-enriched light in the morning and evening, then measured insulin resistance. Morning exposure increased insulin resistance by 30%. Evening exposure increased it by 19%. Both effects occurred independently of sleep duration or caloric intake. The mechanism is direct. Blue light suppresses melatonin, which disrupts circadian regulation of glucose metabolism. Insulin receptors lose sensitivity. Glucose lingers. The pancreas compensates by secreting more insulin. Over time, the system recalibrates to a higher baseline. What was once abnormal becomes the new normal. A 2019 clinical trial published in Investigative Ophthalmology and Visual Science tested blue-blocking glasses worn for 1 month in the evening. Participants showed measurable improvements in fasting plasma glucose, insulin resistance, and sleep quality. The variable was light. Remove the blue. Restore the melatonin signal. Fix the metabolic timing. Immune cells operate on circadian schedules. Natural killer cells, T cells, and B cells all exhibit time-of-day variation in activity. Melatonin is the conductor. A 2024 study in PMC examined how melatonin enhances NK cell function in aged mice. NK cells patrol tissues, identify virus-infected cells and nascent tumor cells, and kill them on contact. Melatonin increased NK cell number, maturation, degranulation capacity, and interferon gamma secretion. It activated the JAK3 STAT5 signaling pathway, which drives expression of TBET, a master transcription factor that controls immune cell differentiation and cytotoxic function. When melatonin is chronically suppressed, NK cell activity declines. T cell function weakens. Immune surveillance becomes less effective. Pathogens gain extra replication cycles. Abnormal cells escape detection. A 1995 review published by the National Institutes of Health noted that chronic melatonin administration substantially increases NK cell activity. The converse is also true. Chronic suppression leads to immunocompromise. You do not just get sick more often. Your body loses the ability to detect and eliminate threats early, before they become systemic. You do not collapse. You remain functional. You work, drive, parent, perform. Coffee smooths the edges. You attribute the fatigue to stress, workload, or age. But beneath the surface, the ledger is being written. Charles Cheisler at Harvard Medical School and Brigham and Women's Hospital calls this circadian misalignment. It occurs when the homeostatic drive for sleep conflicts with the circadian drive. By 11 p.m., after 16 hours of wakefulness, your homeostatic pressure is enormous. Your brain is chemically saturated with sleep-promoting signals. You are exhausted. But if you spent the last 2 hours staring into a screen emitting 460 nanometer light, your circadian system received a different message. It is still day. Melatonin secretion was suppressed. The internal broadcast is still alert, produce, respond. Eventually, exhaustion wins. You fall asleep. But the sleep you enter is biologically mistimed. Cortisol remains elevated longer. Growth hormone is blunted or delayed. The glymphatic system activates late and works in compressed time. You log 8 hours in bed, but the actual duration of the biological night, the window when melatonin was high and repair systems were fully active, was only 6 hours. The factory kept machines running at full power while the maintenance crew tried to replace parts. Repeat this for a decade. The tolerances do not shift back. They degrade. From your perspective, you slept fine. You were unconscious for 8 hours. From the body's perspective, cortisol stayed elevated 90 minutes longer. Growth hormone release was 40% lower. IL-6 and TNF-A remained above clearance threshold. Glymphatic clearance of beta-amyloid was compressed into 5 hours instead of 7. DNA repair pathways operated under time pressure, leaving more unresolved lesions. NK cell reactivation was incomplete. The body does not count hours, it counts repair cycles completed. It measures waste cleared, proteins folded correctly, lesions repaired, inflammatory signals resolved. You felt okay. The cellular accounting says otherwise. Melatonin evolved to be the temporal authority. It divides the 24-hour cycle into two distinct operational modes, day and night. Modern life severed that authority. Biological systems compensate. Compensation is finite. Insulin receptors lose sensitivity 1 percentage point at a time. DNA repair systems leave 1 extra lesion unresolved per night. The glymphatic system clears 2% less waste per cycle. NK cells miss 1 additional abnormal cell per patrol. These are not catastrophes. They are micro-failures, but they accumulate. After 10 years, fasting glucose is 10 mg/dl higher. Inflammatory markers are persistently elevated. Cognitive processing is measurably slower. Immune response to vaccination is weaker. After 20 years, prediabetes becomes diabetes. Chronic inflammation becomes autoimmune dysregulation. Mild cognitive impairment becomes measurable neurodegeneration. Immune surveillance failure becomes cancer. Melatonin suppression does not cause fatigue. It removes temporal authority from the organism. Systems without temporal authority degrade faster. This is not lifestyle. This is lifespan.